Welcome back to Acing AP Bio. In this video, we will explore the structure and function of enzymes and their role as catalysts in organisms. In the last video, we discussed activation energy and how even spontaneous reactions need energy input in order to proceed. Now let's look at an overview of enzyme function. Catalysts speed up chemical reactions by lowering the activation energy barrier so the reaction can take place more easily. Enzymes are biological catalysts made up of protein and have unique three-dimensional conformations. They are very specific to the reaction that they will catalyze. Enzymes are very effective, especially because they are not used up in chemical reactions. This energy diagram displays both an uncatalyzed and catalyzed reaction. Notice that the catalyzed reaction requires less activation energy because the enzyme lowered the activation energy barrier. It is important to understand that the enzyme does not change delta G, which indicates the amount of free energy released in exergonic reactions or absorbed in endergonic reactions. The enzyme has no effect on the amount of reactants and products, but just increases the rate of reaction. Now let's look over enzyme structure. Enzymes are specific to the substance that can bind to it, and this is due to its unique three-dimensional shape. The substance that binds to the enzyme is called the substrate. When the substrate binds, the enzyme catalyzes its transformation to a product, and then the product is released. The area to which the substrate binds is called the active site. It can hold two or more reactants. The shape of the active site is compatible with the specific substrate that can bind to it. When the substrate binds to the active site, the enzyme changes shape so that its chemical groups are more effectively interconnecting with the substrate, forming weak bonds like hydrogen bonds. This slight change in shape of the active site is called the induced fit. The induced fit allows the enzyme to lower the activation barrier because the induced fit helps the enzyme orient the substrates correctly, putting stress on the substrate bonds, allowing the bonds to break more easily, quickening the rate of reaction. The active site also creates a microenvironment specific to the substrate's most efficient conditions. For instance, for a reaction that takes place well in a pH of 4, the active site of the appropriate enzyme will match that pH, allowing the reaction to take place more easily. Next, let's discuss enzyme saturation. The greater the amount of substrate, the more quickly the reaction takes place. However, when substrate reaches a certain concentration, the enzyme can become saturated with the substrate, indicating that all the active sites of the available enzymes are holding substrate. At this point, the reaction becomes constant. There are several factors that affect enzyme function. The enzyme's environment must be conducive to its conformation. An increase in temperature increases the kinetic energy of molecules, so the amount of molecular collisions go up, increasing the enzymatic reaction rate. However, if the temperature gets too high, the overload of kinetic energy causes disorder among particles and destabilizes the enzyme structure, leading to the denaturation or the loss of the enzyme's native conformation. A loss of structure leads to a loss of function. Enzymes also have optimal pH ranges. Most enzymes function best in a pH of 6 through 8. However, enzymes in the stomach have an optimal pH of 2. Salt concentration also impacts enzymes. If salt concentration grows too high, the ions will disrupt enzyme structure, and thus the enzyme will become dysfunctional. There are also substances called inhibitors that inhibit enzyme function. Inhibitors can bind irreversibly with covalent bonds, like the poison cyanide, or they can bind reversibly with weaker bonds. There are two types of inhibitors: competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are substances that are similar to the substrate in shape, so they can bind to the enzyme's active site, thereby preventing the substrate from binding. Non-competitive inhibitors bind to another part of the enzyme, not the active site. This binding induces an enzyme to change its form, so the substrate no longer fits in the active site. Several antibiotics work this way. Although inhibition can be detrimental at times, the selective repression of enzyme activity in the cell is vital for regulating metabolism. Now let's discuss the enzymatic involvement with the regulation of metabolic pathways. We will discuss three types of regulations: allosteric regulation, feedback inhibition, and cooperativity. Allosteric regulation results in the impediment or promotion of enzyme function. The allosteric site is a site for a receptor that is different from the active site. The allosteric site is commonly found between subunits of an enzyme. Allosteric enzymes have two different formations, one that is active and one that is inactive. When a substance known as an activator binds to the allosteric site, it generates the enzyme's active formation and stabilizes the enzyme. When a substance known as an inhibitor binds to the allosteric site, it acts as a non-competitive inhibitor and engenders the inactive form of the enzyme. Feedback inhibition is very common. It occurs when the final product of the metabolic pathway acts as an allosteric inhibitor for an enzyme that regulates an early step of the pathway. This allows the cell to preserve resources. In this diagram of a metabolic pathway, each letter represents a different step of the metabolic pathway, regulated by 4 different enzymes. The final product, E, acts as an allosteric inhibitor of enzyme 1, inhibiting its activity and stopping the pathway. Cooperativity is a form of allosteric activation where substrates enhance the active conformation of an enzyme. It occurs when a substrate binds to the active site of one enzyme subunit, which generates a shape change in the other subunits, making them more active. In fact, they become locked in their active state, and so they are prevented from switching back to their inactive state. Enzymes are so important for life. I hope this video helps you better understand their vital functions in organisms. In the next unit, we will explore the process of cellular respiration. See you then. For more bio content, hit subscribe.